we discussed about microorganisms. There are different variety of microorganisms, as some of example bacteria, yeast, mold, protozoans, and so on. And we discussed previously about the benefit and the harmful effect of those microorganisms. Finally, we discussed about the germ theory of disease, known as spontaneous generations and non-spontaneous generations. But in this week, in week 2 of term 3, we are going to discuss about a lot of things. Let's say the objective of this today's lesson is, at the end of this lesson, the students are expected to list the systems of microbial control, again explain the techniques of microbiological control. Let's continue one by one. And the next part is one of the main core of the topic is microbiological technique. This type of technique is a system of controlling microorganisms, or it is a way of preventing the spread of microbes that leads to a disease, and also it's a method of reducing the number of microorganisms. This is one of the technique mainly applicable in the microbial body. So there are different mechanisms or techniques that are used to prohibit or inhibit the growth of microorganisms. The first one is disinfection, next sterilization, and finally pasteurization. Dear students, let's discuss one by one. The first type of microbial control is disinfection. In this topic, or in this part, we are going to discuss the meaning of disinfection and what are those disinfectants or chemicals that are mainly applicable for this process. Disinfection is the mechanism of elimination of pathogens or disease-causing microorganisms by using disinfectants. So called, as example, local areke or bleaching methods or bleaching agent, calcium hypochlorite, formalin, sodium hypochlorite, and so on. Those are one of disinfectants mainly applicable. Those disinfectants are chemicals that are used to kill the microbes on inanimate surface, whereas the opposite of disinfectant is antiseptics. Antiseptics are applicable on living tissue to stop the multiplications of or the growth of microorganisms. So the main difference between disinfectant and antiseptics are the surface where they are applicable. Disinfectants are applicable on non-living surface, whereas antiseptics are applicable on animate surface. Let's see some of examples of antiseptics like gentian violet, alcohol, areke, ethanol, or hand sanitizer. Now, at this time, we are going to use for coronavirus or COVID-19 hand sanitizer. This is one of antiseptics mainly they are used in our hands. And the effectiveness of disinfectants mainly depends upon one, the resistance of microbes. There are some microbes that have endospores or spores. If they have spore, they are capable of resisting disinfectant, time, pH, power of hydrogen, temperature, and the concentrations of chemicals, where firstly the chemical is manufactured in the industry. Those are the main criteria or requirements that are depends the disinfectant effectiveness. Where those disinfectants are applied, disinfectants are applied in home to clean the kitchen. They are applicable in the launders for clothes. What else? They are applicable again in the dairy and the food industries, on the utensils, on the floor and the rooms, and the toilet, the drinking water. Again, milk containing keepsinks, they are applied mainly in the food technologies industries. Remember that we cannot use disinfectants on people. Students, there are a lot of characteristics about disinfectants. Disinfectants are fastly acting. They are easily penetrate material without discoloring. Disinfectants are effective against all type of infectious disease or infectious agent without destroying the tissue, and they are easy to prepare and stable on exposure to heat and sunlight. And also they are mostly unpleasant, not unpleasant to work with. This is one of some of the pictures that shows disinfectant or chemicals which are taken from your textbook. The second type of microbial control is sterilization. I hope you are with me. What's sterilization? Sterilization is the total destruction or the total removal of microbes. There are different methods of sterilization. These are, the first one is autoclave. Autoclave sterilization is a type of sterilization that can kill a bacterial spore, including endospores, by raising temperature. That means that we can raise the boiling bacterial spore in the water at 21 degrees Celsius at a very high pressure for 15 up to 45 minutes, which means in average 30 minutes we can boil the spore of bacteria in the water with a high temperature, with a high pressure. Let's say some of a bacteria that contain endospores are Clostridium bacteria, which leads to Clostridium tetani. Clostridium tetani is a causative agent that leads to tetanus. Hepatitis virus, those have spores that are capable of resisting high temperature. I remember that water boils at 100 degrees Celsius for 10 minutes. When you can compare autoclave sterilization with that of the boiling point of water. The second type of sterilization is dry heat sterilization. This type of sterilization of bacterial spore for a higher temperature and for a longer time, which is directly proportional. Time and the temperatures are directly proportional. This is why bacterial spores does not die at lower temperature. Dry heat sterilization is applicable at 160 degrees Celsius for 2 hours. It is advantageous type of sterilization due to a non-rusting of instruments. The third type of sterilization is moist heat sterilization. Moist heat sterilization is killing the vegetative part of microbes by boiling 60 degrees Celsius for 5 to 10 minutes in water. Moist heat. This type of sterilization is disadvantageous due to the resistance of endospores of bacteria. This means that the temperature is low. Again, it is the time taken to kill bacteria is also small. So this is disadvantageous type of sterilization. The fourth type of sterilization is irradiation type of sterilization. You can killing a UV sensitive or ultraviolet sensitive spores mainly found in the rooms. So this is mainly by having a natural source, sunlight. And the last type of sterilization is chemical sterilization. We can use chemical sterilization on the food, on the instrument and apparatus using chemicals like ethylene oxide. This is one of all about the second type of microbial control. Students, now we come to the last topic of this lesson. The third type of sterilization, type of microbial control, is pasteurization. Pasteurization is a method of killing pathogens by heating for a certain time, which is followed by rapid cooling. This is originally designed by Louis Pasteur, one of scholars. It prevents the milk and the wine from becoming sour. This type of method doesn't kill all bacterial cells because the inactive bacterial cells with spores will survive and will be able to grow later. Dear students, now we finished today's lesson. Today I think we are going to discuss about a microbial control. There are 3 main microbial controls, which are disinfection, sterilization and pasteurization. The target of these all microbial methods are used to inhibit or reduce the number of microorganisms. This is all about. So if you have any question, you can ask.